This is Rethik, your biology subject matter expert from MEMONEET. In the last video, we have gone through prokaryote or prokaryotic cells, right? So today we will be concentrating more on eukaryotic cells. What are eukaryotic cells and what are the organelles that comes under eukaryotic cells? So what do you mean by eukaryotic cells here? They are the cells that contains a membrane-bounded nucleus and other organelles. So eukaryotic cells have a nucleus which has been enclosed with the nuclear membrane, very important point, and they actually form what? A large and complex organisms, right? So basically, in what kind of complex organisms you can see this nuclear membrane, nucleus, and all. So, such as protozoans, fungi, plant, animals, and all will be having what? Eukaryotic cells. So, the genetic material, like because from there, the genetic material, which is actually called as chromosomes, is being, present inside the nucleus. So they are being formed inside the nucleus also. Say like if we have to concentrate more on animal cells as well as plant cells. Okay. So what one thing we can have a rough in mind is plants and animal cells are different as form. They have a cell wall, different cell wall is present. Then their, central vacuoles will be large, and whereas in animal cells, the central vacuole will be absent, then plasmids will be present. And on the other hand, whereas in animal cells, you will see actually what? They have centroids will be present, which are absent in the plant cell. So, like, this is the major differences that we observe in animal cells as well as plant cells. Then the very first thing that I was speaking about is cell membrane. So what is the cell membrane? So a very first model which has been presented based on the cell membrane is the fluid mosaic model, which was proposed by S.J. Singer and Graciel uh, Nizolans. So what the model says, or what exactly this model explains, it explains the structure of the plasma membrane of the animal cell as a mosaic of components, such as that means mosaic of components in cells. It consists of one thing is phospholipids, then it also consists of proteins, cholesterols, and carbohydrates. This is very important. These are the compositions which plasma membrane consists of. So these components, it actually gives a fluid character to the membrane. Okay. So this is how initially you will see the structure. Say like each. So if this is a cell, right? Obviously, if this is a cell, you will see this kind of membrane now. Okay. Not only on the cell surface, but also on the organelles also you will be seeing nuclear cell wall, everything. So each phospholipids has hydrophilic head. Say like if this is the phospholipids, okay, if this is the phospholipid, it is going to have this hydrophilic head region. Okay, this is hydrophilic head group, and another one will be hydrophobic tail. So this hydrophilic head region will be pointed outwards the environment, okay, outside, pointing outside. And hydrophobic tail that you are seeing will be, formed inside the bilateral. If this is the bilateral, the head region that is hydrophilic head region will be outside and hydrophobic tail will be inside. So in human beings, the membrane, the cell membrane also possesses what? So these membranes possess one of proteins, carbohydrates also, and the ratio of the proteins and lipid varies constantly in what type of cell they are. Say like in human beings, the membrane of erythrocytes have approximately 52% of protein and 40% of what? Lipids. Then plasma membrane. Say like the membrane protein can be classified as two. One is integral protein and peripheral. Okay. So what is peripheral or integral protein? That thing we will just concentrate. So are you seeing here? So this is an integral protein. So Peripheral protein lies on the surface of the membrane. So whatever the proteins here you will be seeing, right? These proteins, they are what? Peripheral. Okay, peripheral proteins. They normally lies on the surface of the membrane, while this integral protein, proteins are partially or totally buried in the membrane. So these integral protein will be partially or completely they will be buried inside the membrane. So one of the most important function of the plasma membrane is, what is the major important function of the plasma membrane? Of course, protection, give the proper cell structure and all. One thing, transport of the molecules across it is very important. Can you see this graph or video where you will be seeing what? A transport of molecules through this plasma membrane, across the plasma membrane. So the membrane is, in that way, one thing is clear, that the membrane is selective permeable to some molecules. That means they will allow only few molecules to pass through them. Say like some specific cells plasma membrane, they are only will allow their specific molecules to pass, not any other such molecules or anything. Okay. So from one side to the other side, or one end to the other end. So many molecules can move briefly across the membrane without any requirement of energy, and this is actually called as passive transport. So passive transport, in sense, it happens naturally without an initiance of energy and all. The molecules will pass from one side to the other side. Whereas in active transport, there is again a requirement of ATP is required to transport the proteins or molecules from one place to the one side to the other side of the membrane. So neutral solutes may move across the membrane by process of simple diffusion method along with the concentration gradient. So that is from higher concentration to the lower concentration, say like salts, some of the ions and all. So that movement can be absorbed here. So water may also move across this membrane from higher to the lower concentration. That is what they're saying. So movement of water by diffusion is actually called as osmosis, as the polar molecules cannot pass through the non-polar lipid bilayer. So they require a carrier protein of the membrane. Say like, so there are some polar molecules, they will unable to, pass through the non-polar lipid bilayer. Certain restrictions will be there. So that time they required some carrier protein of the protein for the membrane to facilitate this transportation across the membrane. Then a very few ions or molecules are transported across the membrane against their concentration gradient for lower concentration to higher concentration, vice versa. Such a transport is actually where you require active, transportation. That was which I was initially saying in the previous slide, right? So that comes here. So for the transport of energy dependent process, like which ATP is completely utilized, so that thing we actually called as active transportation, like sodium pump channel, then potassium pump channel and all is there, right? So they initially are active transportation. Then what is the cell wall? So can you see this? Yeah. So this is what? Cell wall. So non-living rigid structure, they are actually called as cell wall, forms the outer coverage of what? The plasma membrane. So it could be fungi, plant, animals, whatever it is. So cell wall not only gives a shape of the cell, but also protects the cells from any mechanical damage and infection. This is basic thing. It also helps in cell to cell communication or interaction, and it also provides a barrier to undesirable macromolecules. That means it acts as a barrier. It will not allow any undesirable macroorganisms to penetrate or move. Then we have also algae, which have a cell wall, which is made up of what? Cellulose, then galactans and mannose and minerals like calcium carbonate. While in plants, if you see the composition of the cell wall, you will initially see what? You will see the young plant cell. A primary wall is capable of growth, which gradually, it gets matured. Cell gets matured and reaches to secondary wall as it forms on inner, like towards membrane side of the cell. Then we have middle lamellae, right? So what is this middle lamellae? Is a layer mainly of calcium peptide, which holds or uh, glues the different neighboring cells together. then the cell wall and middle lamellae may be transferred by plasmo. So they will be transferred. Okay, that means the cell wall and the middle layer, whatever the molecules they come, so they may be transfers by the plasmo desmate, which connects the cytoplasm and the neighboring cells together. So the next topic is endoplasmic reticulum. So it is again an ordinal. So when you see or when you study any prokaryotic cells, okay, under the electronic microscope, so you will actually see the presence of a network or reticulate of a tiny tubule-like structure which is scattered in the cytoplasm. So we normally call them as endoplasmic reticulum. So here this endoplasmic reticulum has been divides the intracellular space in two distinct compartments. Say like inside the endoplasmic reticulum, we normally call it as lumen, and outside, that is extra lumen, which is the cytoplasm compartments. So one thing that you're going to see here is we have endoplasmic reticulum often shows this ribosomes. Okay, so there these ribosomes that are attached onto the outer surface of the endoplasmic reticulum. So those makes those surface very rough one. So we normally call it as rough endoplasmic reticulum. Okay, so if there is an absence of these ribosomes onto the surface, then obviously the surface will be smooth, and we name that as smooth endoplasmic reticulum. So this rough endoplasmic reticulum is frequently absorbed in the cell activities that actually involves in the synthesis of proteins and secretion of the proteins. So they are extensive and continuous with the outer membrane of the nucleus. And coming to the smooth endoplasmic reticulum, it is the major site of the synthesis of what? Lipid. Okay. So for protein synthesis, you will see rough endoplasmic reticulum. For smooth, you will see lipid synthesis. So in animal cell, lipid like certain hormones like steroidal hormones, they are being synthesized by this smooth endoplasmic reticulum. So the next one we have is the Golgi apparatus, which was first absorbed by the Camillo Golgi in 1898. So he did a densive staining reticular structure near the nucleus. So these were later named as Golgi bodies. Okay, after him. So they consist of many flat disc-like, shape structure which measures of 0.5 meters to 1.0 meter in diameter. So these are stacked parallelly to each other. That means, can you observe here? They are very parallel to each other. They are being joined. So various number of cisterns are present. So these are actually called as cisterns. So they are present in one cell to the other Golgi complex differs. So they are concentratedly, they are arranged very nearer to the nucleus with a distinct convex cis or the forming phase and concave trans or the maturing phase. That is how they are nearer to the nucleus. Then the cis and the trans phase, like inner and outer phase of these organelles are entirely different, but they're interconnected. So their function, everything is different, but they're always interconnected. So the Golgi apparatus principally perform the functions of what? Major function is packing materials to be delivered either to the intracellular target or secreted outside the cell. So they acts as a vector or a delivering agent. Okay. So they pack up the proteins or whatever the targeting, hormones and all, and they'll go to the targeting sites. So materials to be packed in the form of vesicles from the uh, endoplasmic reticulum fuse with the cis phase of the Golgi apparatus, and it is going to move to the matured phase. If it is a cis phase, so it will pack, then later it will go to the matured phase. This explains why the Golgi apparatus remains in a close association with the endoplasmic reticulum. So the number of proteins synthesized by the ribosomes on the endoplasmic reticulum, so they are being modified in the strain of the Golgi apparatus before they are being released from the trans phase. Okay. So that means if there is anything that they need to be rectified and all, the proteins will be modified in the strain of the Golgi apparatus before they are being released from the trans phase. Golgi apparatus is an important site for the formation of two things. One is glycoproteins and glycolipids. Clear? The next two you will see is lysosomes and vesicles. So then what do you mean by lysosomes here? So they are the membrane bounded vesicle structure which are formed by the process of packing of the Golgi apparatus. So the major thing is the isolated lysosome vesicles have been found in a very rich in almost all types of hydrolytic enzymes, okay, such as hydrolysis. It is going to hydrolysis is nothing but it is if these are some enzymes or some proteins, so it is going to lysis, it is going to cut them, okay, into small tiny pieces. So hydrolyzed lipids, protease, carbohydrates will be there. Then optimally they're active at high acidic pH. That means they are very active at optimally active at the acidic pH. So these enzymes, they're capable of what? They're actually capable of digesting the carbohydrates, then proteins, lipids, and nucleic acids. So this is the major function. The next we have is vacuoles. So it is the membrane bounded space which is found in the cytoplasm. So these vacuoles, contains what? You'll be seeing water, sap, excretory products, and other waste materials which are not, useful for the cell. So the vacuoles is bounded by a single membrane. They are called as tonoplast. This is very important. So here you are seeing here the single membrane, which is actually called as tonoplast. Very important with the neat aspects. So in plant cells, the vacuoles can occupy up to 90% of the volume of the cell. Whereas in plant, the tonoplast facilitates the transport of number of ions here and other materials against the concentration gradient in the vacuoles. So therefore, here you can concentrate. You can see here, the concentration is significantly. It will be obviously high in the vacuole than in the cytoplasm. So say like if you consider amoeba, the concentrative vacuole is important for excretion. So in many cells, that means the waste all it has to extrude from the body. So in amoeba, the vacuoles play a very important role. So in many cells, as in protease, food vacuoles are formed by engulfing the food particles. So this is something basic. So in the next video, we will be learning the remaining part that are like nucleus and the DNA structure, chromosome and all mitochondria. Yeah.